Hello students, I welcome you all in this e-lecture on the topic Role of Metal Ions in Biological System. So in the previous lecture, I told you about the introduction to the essential elements in our biological process. So here you can see on the screen that the elements which are known to be essential for human life are shown in purple color, and as you have seen that 19 of these elements are quite essential for humans. In the previous lecture, I gave you the criteria for the essential elements, that a physiological deficiency appears when the element is removed from a purified diet. So this is known to be an essential one for the body. And the deficiency can be relieved by the addition of one specific element. And the third criteria was that a specific biochemical function is associated with a particular element. हर एक एलिमेंट के साथ कोई ना कोई एक बायोकेमिकल फंक्शन जो है वो जुड़ा हुआ है, विच इज़ क्वाइट एसेंशियल फॉर द फिजियोलॉजिकल प्रोसेसेस टू बी कैरीड आउट. So the simplest definition of an essential element will be what? That it is an element required for the maintenance of life. Its absence results in death or a severe malfunctioning of the organisms. So experimentally, this rigorous criterion cannot always be satisfied, and this has led to a broader definition of essentiality. So now we come to what? That an element is considered essential when a deficient intake produces an impairment of function, and when restoration of physiological levels of that elements relieves the impaired function or prevents impairment. So the organism can neither grow nor should have nor it completes its life cycle without the element in question. It should have a direct influence on the organism and be involved in its metabolism. And furthermore, it should be noted that the effect of the essential element cannot be replaced by any other elements. So we also went through that what are essential elements, and further they were classified into two classes, macronutrients and micronutrients. So this was all about the introduction of these metal ions in the biological processes. Now we come to the functions of these metals in our body. As you know that metals are commonly found as natural constituents of proteins. Protein का एक inherent part होते हैं metals. So nature has learned to use the special properties of these metal ions to perform a wide variety of specific functions associated with life processes. So in this regard, I would like to first come to the role of metal ions dioxygen transport. So as you know that one function uniquely performed by a metalloprotein is respiration. Okay, so as you know that there are three known classes of dioxygen transport protein: hemoglobin, myoglobin family, hemocyanin and hemoerythrin. And in the coming screen you will see the functional metallic cores of these proteins. In these proteins, a delicate balance is achieved whereby the oxygen molecule binds to an iron or copper center without undergoing an irreversible electron transfer or redox reaction leading to oxygen-oxygen bond cleavage and concomitant oxidation chemistry. Okay, तो hemoglobin and myoglobin में आपको पता ही, you see this structure. This is the structure of hemoglobin with and without oxygen. So the function of oxygen transport and storage in higher animals, as you know, it is provided by iron porphyrin complexes, which are hemoglobin and myoglobin. तो hemoglobin जो है, it transports the oxygen from lungs to site of its use inside the muscle cell, and there the oxygen is transferred to myoglobin for use in respiration. The iron in hemoglobin and myoglobin is in +2 oxidation state. So this oxygen molecule, it first coordinates to iron 2 from one end of heme, and globin protects the iron 2 to be oxidized to iron 3. ठीक है ना? तो as you see on the screen, so this oxygen molecule, when it is getting coordinated to iron 2, and globin is protecting the iron 2 to be oxidized to iron 3, what it does, it creates a hydrophobic environment so that the water molecule could not reach the site. Assuming there is no globin present, the oxidation reaction would have occurred and the product hemetin, in which iron is in +3 oxidation state, must form. Hemetin cannot bind oxygen, thus becoming useless for oxygen transport. तो ये देखिए, this is the structure. Now we come to these that the structure of the porphyrin ring and some other organic cofactors have been shown here in the coming figures. The other two respiratory proteins employ a pair of metal ion instead of one. देखिए hemoglobin और myoglobin में तो एक आयरन एटम था, जबकि दूसरे हेमोसाइनिन में जो है दो there are a pair of two metal ions. देखिए इस पे you see in the hemosainin there are two copper ions. So this is again the figure which is depicting the hemosainin, structure of hemosainin with and without oxygen. And then you have the structure of deoxyhemerythrin and oxyhemerythrin. So these are the functional units which are found in the three classes of dioxygen binding protein. In hemerythrin, you know, which is a non-heme oxygen binding iron protein, there are two iron atom here. So it is used in great variety of marine worms and phyla of marine invertebrates. So it contains iron 2, which binds oxygen reversibly, and when oxidized to iron 3, methemerythrin is formed, which shows no tendency to bind oxygen. Okay, so this we will study in details in the coming lectures. And similarly, in the hemosainin, you can see that it contains a pair of copper atoms. There are two copper atoms, and it binds one molecule of oxygen for every pair of copper one ion. When oxygen is bound to hemosainin, its color is blue. Otherwise, unbound hemosainin is found to be colorless. They are found only in mollusks and arthropods, and they occur dissolved in blood and hemolymph, and usually comprise of nearly 95% of the total protein present. Each subunit contains two atoms of copper and polypeptide chains. These subunits, they further form aggregate with a molecular weight up to 9 to 10 to the power 6. So these are the protein molecules which we will take in the coming lectures in details. Here you can see the structure of heme protein. So this is all about the hemosainin and hemerythrin. Now we come to the electron transfer processes, that is the role of a metal ion in electron transfer processes. So the dioxygen binding metal center in hemoglobin illustrates how bioinorganic chemistry uses one of the major classes of chemical reactions, that is acid-base chemistry. As you know that a Lewis base oxygen binds reversibly to a Lewis acid, which is the porphyrin bound iron atom. Another main class of chemical reactions, those involving net electron transfer, occurs in proteins that undergo redox transformation without themselves catalyzing an overall chemical change in a substrate molecule. So such bioinorganic current carriers usually pass their electrons to and or from enzymes that require redox chemistry in order to perform a specific function, for example nitrogen fixation. Sometimes the redox active center is directly incorporated into the metalloenzyme, and we know that the two electron transfer centers widely encountered and experienced in biochemistry are iron sulfur clusters and cytochromes. So first you should have an introduction about the cytochromes. Cytochromes क्या हैं? They are heme proteins, those acting as electron carriers involved in photosynthesis. आपने अपने प्रीवियस क्लासेस में भी फोटोसिंथेसिस के बारे में पढ़ा होगा, तब यू मस्ट हैव कम अक्रॉस दिस टर्म cytochromes. It consists of a porphyrin ring chelated to iron atom. These cytochromes are found in all aerobic form of life, and here the oxidation state of iron may either be 2 or 3, and they exist as redox intermediate in electron transfer reactions. इन सबको भी हम आगे आने वाले चैप्टर्स में पढ़ेंगे. They are not only present in chloroplast for photosynthesis but also are present in mitochondria and take part in reverse process of respiration. It is important to note that iron functions well in cytochromes because iron 2 and 3 redox EMF has a value in the appropriate range for life processes. देखिए, जैसा कि मैंने आपको बताया कि नेचर किस तरह से जो इन मेटल आयंस का जो इनकी कैरेक्टरिस्टिक प्रॉपर्टीज़ हैं उनको लाइफ प्रोसेसेस को कैरी ऑन करने के लिए हाउ ब्यूटीफुली इट डज इट, how beautifully it utilizes their chemical properties. So now we come to the iron sulfur proteins. They are also another one class which are involved in electron transfer reaction, which are acting as electron carriers. Several non-heme iron sulfur proteins are involved in electron transfer. They are made up of peptide chains having cysteine. Cysteine moiety जो है amino acid होती है, where cysteine sulfur is coordinated to iron or cluster of iron sulfur atoms. So the term rubidiroxine is used for those having one iron atom, okay, while those containing iron sulfur clusters they are called ferridoxine. तो rubidiroxine and ferridoxine के बारे में भी हम आने वाली क्लासेस में डिटेल्स में पढ़ेंगे. Now you should know that ferridoxine is the first electron acceptor in photosynthesis. And then we have blue copper. It also acts as an electron carrier in photosynthesis and mainly occur in algae, green leaves and other plants. And the different form of blue copper are azurin, plastocyanin, stellocyanin, etc. So this was all about the role of these metal ion in electron transfer processes and as dioxygen transport in body. So in the next lecture we will go through their role in other processes, other life processes. Till then, goodbye.